This is Kimberly.
This is Michelle.
Today we're going to be going through the stages of the cell cycle, mitosis, describing chromatid and chromosomes, and taking a closer look at cancer.
Part 1, the cell cycle. The cell cycle is what we call the several processes that cells experience throughout their lifetime. It's divided into 4 phases, G1, S, G2, and M phase. Although sometimes this part right here, the interphase, it's considered a phase resulting in 5. And the interphase consists of the G1 phase, S phase, and G2 phase. It's a term used to refer to the stages with no cell division. So anyway, during the G1, gap 1 phase, this is where the cell grows and develops the most. The cell spends time growing in size and making new proteins and organelles. Then the S phase synthesis starts, and DNA is synthesized along with proteins required for chromosome replication, and the chromosome makes an exact copy of itself right here in this phase. And then it brings us to the shortest phase, gap 2. In this phase, the cell basically checks over what the organelles it still needs and makes those organelles and prepares for the M phase, which is right here. And the M phase is where the cell division happens. It's not a part of the interphase, which you can tell clearly. And then in eukaryotes, it consists of 2 main parts, mitosis and cytokinesis, but we'll get back to that later.
Part 2, chromatin, chromatid, and chromosomes. We've all heard of DNA and stuff, but it's not that simple. DNA is arranged into structures called chromosomes that are made up of nucleic acids and proteins. That's pretty simple, except that there's also chromatins and chromatids. A chromosome is what you call the DNA during mitosis, when it's become compacted and visible. The chromosome is usually represented by an X at mitosis. At the center, there's a centromere that holds the 2 sides together. These sides are called sister chromatids. They're identical as they were produced during replication at the S phase. Before the S phase, this DNA was barely visible and in very loose strands called chromatids. During the S phase, the chromatins get condensed and wrapped around proteins called histones. They then form chromatids, which then form chromosomes. Basically, before the S phase, the DNA is chromatin. During the S phase, it becomes chromosome. The chromosome is made of 2 sister chromatids, however, which is important during mitosis.
Part 3, the M phase, mitosis and cytokinesis. Mitosis is when the cell's nucleus divides into 2 daughter ones with the same chromosomes. It's split into 4 phases: prophase, metaphase, anaphase, and telophase. In this diagram right here, you can also have the prometaphase, but basically it's included in the prophase whenever we're going to talk about it.
It begins at the prophase, which is the longest. The chromosomes condense further during this, becoming visible. 2 little centrioles, my bad you guys, in your cytoplasm, near the nuclear envelope separate to opposite sides. Then they help organize the spindle, a structure that helps separate chromosomes in the centrosome region. The chromosomes attach to the spindle fibers near the centromere, or at each chromatid as the nucleus and nuclear envelope both collapse.
You can tell like the nuclear envelope collapsed because this blue right here, it's not here anymore. But basically, metaphase starts after this, and it only lasts a few minutes. Metaphase is when the chromosomes are moved by the microtubules, this pink stuff, to the center, this spindle equator. And then the microtubules also connect all of the centromeres of the chromosomes to the spindle poles up here and down here. And then after this finishes, anaphase starts.
In anaphase, the centromeres are pulled apart, dividing the sister chromatids that form the complete chromosomes. These chromatids will then be pulled by the microtubules towards one of the 2 poles, forming two groups of daughter chromosomes. When they finally stop moving, telophase begins.
Telophase is the final phase, and it's where the nuclear envelope, this blue right here, it comes back, and then around each group of chromosomes in the spindle, it kind of vanishes. It's not there anymore. And then, while this is happening, the chromosomes of each group also start to decondense back into chromatin, and you can tell by how this is all bundled up, and then this is loose strands again. And then after the cell has finished splitting the nucleus, concluding mitosis. The M phase isn't finished yet, though, because two nuclei doesn't mean two new cells. The cytoplasm still has to split. That's what happens during cytokinesis. And this is where the cell's cytoplasm, the area surrounding it, it splits apart. In animal cells, this part right here, the contractile ring, it pulls the cell membrane in, dividing it into two parts. The parts are similar in size, and they each contain a nucleus, and organelles. But in plants, because they have a cell wall, they can't pull their membrane in, so their cytokinesis is different. In their cytokinesis, the Golgi apparatus sends out vesicles with cell wall molecules in the middle of the cell, and these vesicles form a cell plate, which will split the cell. After it's split, a cell wall will grow back eventually and replace the cell plate. But in both types of cytokinesis, two daughter cells, which are identical, exactly the same, identical, are formed. And this concludes cell division, the M phase, and it also concludes the cell cycle.
That, that was the complete undisturbed cell cycle. Part 4, cancer. Now we're going to talk about what happens when a cell cycle goes wrong and out of control. Cancer is what happens. This is an example of a cancer cell. Basically, as a result of smoking, tobacco, and other things, some of your cells get damaged. These damaged cells will then divide with no restraint and form tumors inside you and your organs. They do this because they're damaged and can't detect the signals that regulate cell growth. So they'll keep growing, or like stacking on top of each other. The mass of detected cells, the tumor, will probably keep growing, potentially damaging your tissues and organs, causing serious illnesses and sometimes death. Basically, don't smoke tobacco and disturb your cell cycle. You're giving yourself cancer and killing yourself. The end.
And these are our sources that we use to get our pictures information.